and I will try to stick to the timeline. It's a great pleasure for me to be here. I see also other associations of patients. So this is an important support for all of us, for those who do research, etc. So we are really glad and happy with that. So we take part with a lot of joy in these meetings. I am a genetist, as you know, and I was asked to talk about the mutational spectrum of this gene as far as we know up to now, and what we know about the interactions between phenotype and genotype. So something that we are going to go through without getting into details. So we don't have a lot of time, but we are going to come back to some of these topics also in the afternoon. So the topic will be further discussed. So very briefly, genetics. As you know, well, maybe these concepts are very simple and easy for most of you, but I thought it was fair enough to talk about that and also to highlight the most important key points. This is autosomal dominant inheritance and disease. The two copies of the gene, only one can be subject to mutation to cause the phenotype, and this has also an impact on the way the disease is transmitted. Well, it affects a lot of younger children, so it's not parents who can transmit this disease. There is, as in dominant diseases, we have a parent who is in fact 50% probability and likelihood to transmit this disease from generation to generation, but for late onset diseases or less severe diseases. In this case, we have an early onset disease, so it's very, very rare that something similar happens. So we have a kind of so-called de novo mutation. Parents don't have it, but during the synthesis of eggs in this process that is called meiosis, then we have this process of formation, and there is a mistake either in the father or in the mother. That is to say, all the cells of the child will have this mutation, but the mutation is new. That's why de novo mutation. So it's not something available in his or her parents. So this is a copy, and here we can find the copy that was subject to mutation. This is something normal. Our DNA is not perfect. When it comes to the replication of DNA, we have some mutations. Sometimes they are just casual. We have 40, 80 de novo variants and mutations that are the result of this meiosis process. So all the gametes that generated us. And in this case, we have benign variants, so no negative impact on DNA, or maybe they are only harmful if there is another copy that was subject to mutation on the other side, so a regressive or recessive state. In this case, we have no disease, but when the variant is ecological and dominant in this case, so it can bring about the disease. In that case, the de novo mutation leads to a child suffering from this mutation. This is something that we saw above all in most cases. So if we do have a de novo variant, we have the recurrence risk in the family, and second brother or daughter that suffers from the same disease. Well, in this case, the risk is extremely low, and this is similar to the one of general population, where there were no children, there are no children, but there is likelihood and probability of having a second child who is affected. Well, in this case, if we have a de novo mutation, the likelihood is very low. But there is another mechanism that was already described for GNAO1, that is this kind of parental mosaicism. What does it mean? It means that parents are not perfectly clean, so to say, but one of them is transmitting this variant or carries this variant, and maybe not all the cells are affected. This was only in a specific group of cells, for example, gonadal mosaism. So in the gonads, so you can carry out perform a blood test, and you can see that there are some precursors of the cells in which you can find the mutation. This does not cause the disease, but the disease is transmitted, is passed to the next generation or to the other people. In this case, you have an example of a mother. In the gonads, there is one copy which is affected. The other ones are standard, normal. Or there could be also a very low level of mutation in the blood cells, but the level is so low that it is not enough to cause the disease. Not 100% of the cells, but maybe 10 or 15% of the cells that are subject to this mutation. We don't realize that, no one realizes that, but there is a risk of transmission to the children, which is much higher. If in one of these cases we can find the mutation in the first child, and then we can verify the same with the parents, just the normal sequencing, standard sequencing method, well, starting from peripheral blood, then we can't find the mutation in the parent. So this is the so-called Sanger sequencing on DNA, and we will not detect the pathogenic variant, and it suggests a de novo occurrence of the affected child. And we have some of these works that can be found here, GNAO1, so sib pairs, so pairs of siblings that have been already reported in these papers, and the recurrence, the risk that is estimated here of having an affected child amounts to 5 up to 15%. So it's not very, very low actually. Well, it's very difficult to analyze gonads, so we have to do this sequencing method starting from the peripheral blood, but nowadays we also have important tools that enable us to assess a very low mosaicism. This is a kind of provocation for all those who deal with the genetics. Sanger sequencing, that goes much faster, but these de novo dominant diseases should be also included into these guidelines, also for parents. So this should be done also for parents, because thanks to this method we can also detect a few mutations, and if we carried out the standard sequencing, we wouldn't see it in a lab. And if we use this different technique, this can be seen much, much better. So a low-grade mosaicism could come up in this case. So this is not something for you. Maybe it's not your cup of tea, but it's a task of those who deal with genetics. So it's something that should be taken into account in order to protect ourselves from or against a risk of recurrence. Well, a couple of general concepts concerning the function of this protein. It's very, very long, actually. This gene encodes the one subunit, alpha subunit of a G protein that exchanges this GDP-GTP. So there is a switch to inactivated to activated. This is a protein in the membrane, and there is alpha and beta and gamma. It can be activated or inactivated if it binds GDP or GTP. So this is a very small, tiny protein, intracellular protein. So if phosphor is hydrolyzed, it can be activated or inactivated. The two complexes are going to detach, and some signals are sent. It's like a triggering factor that, based on the type of cell and on the moment, will regulate a series of pathways, intracellular pathways, that are quite important. This family of proteins to which GNAO1 belongs also has effect on our central nervous system. Receptors of neurotransmitters, for example, adenosine, dopamine, so adrenergic. So there is a lot here. which could be said. Then we also have the main inhibitions, but maybe there are other ones that have not been found out yet. For example, regulation of adenylyl cyclase, and this is a signal of intracellular signal. But also some activities concerning calcium, which are quite important, for example, for epilepsy. And this is for the channels, calcium channels, as Mr. Guerrini said beforehand, and also the release of neurotransmitters through synapses and other proteins for vesicles, and also other channels. And so on. If we meet in a couple of years, once again here, this list will be much longer. We are talking about multitasking proteins that can carry out a lot of different functions. Well, what about the expression of the protein? This is a site that explored the expression of a specific gene in the different tissues of man. So as you can see here, you have the heart, you also have the brain, etc., etc. So the gut, etc. Here we have the brain regions. Well, you can see some peaks, GNAO1 and NAO1 compared to other tissues. So this is something that we can find in the central nervous system, which is very abundant here. So it's the most abundant, the richest one in animals, in mammifers, and this is something that has really a crucial role. If we take a look at this other site, this was issued recently, but it's very nice also here because a study was carried out for the embryo of a mouse, so embryo development in a mouse, not just taking a look at the expression in macro areas like the cerebellum, the cortex, etc., but single cells, so single cell transcriptonic in this case, in which we can see in which cells the protein is expressed during development. As you can see here, we have lots of different stripes and different captions. Even if we can't read, there are a lot of neuronal populations in which this protein is expressed a lot. For example, cholinergic neurons, but also excitatory neurons, interneurons, precursors of androcytes of any kind, the glia, etc., etc. So this is a lot of food for thought. And the phenotype also reflects the expression of this protein. And GDP, something we talked about, so it's a catalytic domain, and then they are rolled out with each other, so there is a long protein with distant points, but after the folding they are attached, they are combined with each other. So you can see here the yellow and the red part. This is the catalytic domain that binds the GDP, and then we will talk about the localization of mutations that leads us a little bit in order to better understand the function. This is the unrolled protein, as you can see, and you can see that the regions that bind this part are scattered on the protein, but then we have a pocket, a single pocket that is set up once again. This is very important because when it comes to mutations, that it can be found on the gene, on the entire gene, so very distant mutations in terms of position, but mutations that can bring about the same phenotype, because when the protein is folded, then they are in contact with each other and they build a specific region with a specific function. So it is important to understand the structure as well as the domain in order to give a sense to the phenotype, genotype interactions and connections. We saw that there were a lot of genotypes, so intellectual impairments, epilepsy, etc., etc. But we have there are two aspects that we should highlight for us and for you. We should try to have predictive factors in order to carry out a prognosis. I always detect mutations, and is it possible, starting from mutation, to understand what kind of disorders they will have, like movement disorders, etc., but also for therapeutical strategies. Here we have a certain number of mutations. We have a block like 50% that is characterized by hotspot mutations, so 246, 203, 209, which are the most represented ones. And then we also have a lot of private mutations that are scattered. Time is running out, sorry, so I'm trying to go as fast as possible. And what about the distribution? Well, we also found some connections in terms of domains, and functions. Well, yes, we have a connection between the phenotype on the one hand and the domain where the mutation can be found. So, for example, the mutations that can bring about movement disorders are within this amino acid range that is outside the GDP binding pocket, while the variants within the catalytic domain cause epilepsy in most cases. So there are some connections between the place on the one hand and the function on the other hand. And then we also have another even more interesting attempt, so characterizing mutations based on their function, NP cyclic in this case, which is one of the most important functions of this protein. Well, a very important aspect that came out is that these have very different behaviors: loss of function or a partial loss of function, so the protein works less or less worse. With the mutation, nothing changes, as if there were no impact. In other cases, we have a gain of function, so the protein works better. And these should also have phenotypical connections, and this is very important. If we take a look at the mutations that can give a movement disorder, are gain of function, as you can see here, or normal function, while the other ones, epilepsies, they are based on loss of function mutations. And these can also be seen when it comes to comparing with statistics. So loss of function, 100% epilepsy, 80% epileptic encephalopathy, and movement disorders 60%, but they are a minor part. And the gain of function is the opposite: movement disorders 90%, epilepsy 50%, and very few have a very severe clinical picture of epileptic encephalopathy, and the other one, normal function, a lot of movement disorders. So we can have a kind of orienteering about that. Concerning the gain of function mutations, there are some very interesting hypotheses. Concerning loss of function mutations, we don't know a lot about them. Time is running out once again, sorry. So I'm going to go very quickly and fast. Well, as far as the pathogenesis is concerned, we have a lot of contacts with other genes, and they will have an impact on similar structures, as you can see here. ADCI5 for this hyperkinetic disorder with the same complex, the same pathway, and then GNB1, GNAL, dystonia, general dystonia, PDE10A. And so the impact also justifies the phenotypes, in some cases with loss of function, in other cases with gain of function. But everything impacts on the NRPC regulation. Well, concerning epilepsy, once again, there are common patterns with a lot of genes that are involved in epileptic encephalopathies and movement disorders, as Mr. Renzo already said, Mr. Guerini, and also calcium channels and forms that are already known. These are only some of the examples we can find, and we can also understand that the gain of function increases the GNB, for example, and this can also bring about disorder of the movement. And in this case, we can see what Mr. Leuzzi has already said: gain of function or normal function. We have both of them. Nevertheless, If we have a normal function on NP cyclic, then the mechanism must be different, because if the pathway doesn't change, there must be something that justifies also the phenotype. And these are other things that we do not know yet, non-canonical pathways that we should take into account. So it's a great pleasure for me to introduce this study, which we are honored to be able to present. Mr. Katanev will talk about that. He's the main author. This is just a review. As you can see here, a very, very simple and easy review, but his work is much more complicated, very, very complex, actually, if you are interested. And it is about the GNAO1 at the level of Golgi and not at the level of the plasma membrane. So we have different places in the cell with a protein that can have also very different functions within the membrane. This protein fulfills all the functions that we saw before, but so epileptic encephalopathies, but the Golgi apparatus, it controls other mechanisms, and it plays a more important role when it comes to the cognitive development and the onset of the movement disorders. So in terms of mutations, if in other cases we do not have anything different here, in this case the cell could be and play a very important role. So what are the take-home messages? We are talking about de novo mutations. This is a good news, piece of news, very low risk, but let's remember the mosaicism that we can have. Also for small labs, we can have a screen, for example, so they can be screened, and then we have a specific clinical suspect. And we start understanding something about the genotype, phenotype correlates, even if they are not that accurate and precise, in order to predict the phenotype in single patients. There is a lot of work to be done. The way is still long, but this is something in order to lay the foundations for you for the development of targeted treatments and therapies. Thank you very much. We're going to move on. Thank you.